All right, before we start, just a few things that I said already 5 minutes back. That Thursday, what we are going to do is we're going to do the exam review, and the honor students are going to put a discussion and one slide on the SARS-CoV-2 and COVID-19 related to this material. I'm going to get those they're going to upload the discussion in the discussion part by Sunday, 11:59, Sunday midnight, and they're going to send me the PowerPoint, and I'm going to just edit the PowerPoint as needed and going to post it on Monday. And next Tuesday, we are going to go over that material. This will be the on-your test, and what we are trying to do in here that conveys some easy to remember take-home message on the SARS-CoV-2 related to this material. And it will not be too technical. It will be something that you will remember beyond the exam, and you can educate the community around you. So that's the purpose. Last time we did it a little bit different, and it was too technical at the honors level, research level. They did really deep dive into the literature, and probably a comment that I get that people did not take home any of those messages. So this time we are doing a little bit different, more of a thing that you can remember. So that's the purpose. And also I said that please come to the class, and I said I just cannot mom you, and I don't. And also just try to take the quizzes pretty seriously. The first time you take will tell you at that time what you know about the material. Depending upon that, usually I do not just pick on people and reach you. If you need any help, please ask me. Again, I'm available during the office hours, out of the office hours by appointment, all the time, through Zoom, nights, weekends, whenever you need help, I can do that. But you have to reach out to me. All right, being said that, let's start the last topic, and you will see that we have 5 topics in here, because the first topic, the topic 1, is very rudimentary. That's the reason that literally the real material is only 4 chapters, and subsequently for other modules we'll have just only 4 of the chapters. You will see that, like 4 topics. So the first thing that, as we know, the viruses are the obligate intracellular parasites, and if you just want to put a molecular parasites, that's more correctly. And the virus particles cannot just diffuse through the plasma membrane, as you know, that because they just can't. They are just too large. They just can't, and they're not soluble in the lipids either, so they cannot go through. So they need to bind to the receptor on the surface of the cell. That's one of the first thing that what happens. And the word that we are used, the surface interactions are multivalent. That means it is just not one hydrogen bonding that is keeping the virus together, or just not one electrostatic interactions that is keeping the virus together, because it's too weak. The viruses are bouncing around. So to gain access to the receptor and take it internalized, there are several interactions in between the viral surface protein and the receptor needed, and that is what we mean by the multivalent interactions. And they may require some passive molecules or receptor. What we will see that what we mean by passive molecules and the receptor in a subsequent slide. Passive molecules are not specific receptors. Like sialic acid, they are present in almost any glycoprotein that is on the surface of the cell. Influenza virus binds to the sialic acid, which is not on a specific receptor, but on many of the glycoproteins. But when they bind to these sialic acid receptor, if they find they will be internalized by endocytosis, we are going to see that. On the other hand, many of the viruses will bind to a specific protein receptor specifically, not randomly to many different proteins. So those are the difference between the passive molecules and the receptor. We're going to see that. And the receptors are the only one upon binding can change signaling, or will basically do a cascade of signaling, and that will be the internalization process. So as you know, that receptor-mediated endocytosis is the most common one. Also, there is a fusion with the plasma membrane, but for the fusion also will need a virus receptor interaction along with the same way. You need the same kind of interaction. You just cannot just take a virus and it will just not readily fuse with the membrane. And the destination of the viral genome is finally the genome is delivered either into the cytoplasm or in the nucleus. And we're going to see that it depends upon the RNA viruses. Many of the RNA viruses, the genome is delivered in the cytoplasm, but some of the RNA virus genome is also delivered into the nucleus. And almost all DNA viruses, almost all, not all, the genome, the nucleus is the destination. Some of the viruses, very large viruses like pox virus, they pack their own DNA polymerases and they can replicate in the cytoplasm. Depending upon what the virus has, the destination depend upon that. And it's very varied. So finding the right receptor, first what happens that as the virus gains entry into the portal of entry, and what is a portal of entry? This is a word you definitely have from your microbiology 3050, from your general microbiology. A portals of entry through which a pathogen gains an entry to a living organism or a body, for us here, human being the host. This could be everything. This could be your mouth. This could be your nasal opening. This could be your urinogenital opening. This could be your broken skin. Sometimes not even broken skin. You can have an intact skin and you have pores through which the virus can gain entry towards the inner compartment. And after it gains to the inner compartment, it just have to be close to a cell which is susceptible, and on those they are just bouncing around. At that time, there are some electrostatic interactions in between the charged amino acids in between the cell surface and those, but they are not ready to be taken inside the cell yet. They're just bouncing around by Brownian motion. But gradually, as they are adhering to the cell surface, they in that process, eventually, they will find a receptor molecule on the cell surface. And more the viruses, chances of finding the receptor is more. So in this process, the receptor is a cellular protein that is destined to do some specific function for the cell. There is no receptor on the cell that is in there to take in a virus. They all have cellular functions. The viruses evolved to exploit them to gain entry into the cell. So that's the main concept of the receptor, that what is the receptor for the viruses. They have all cellular functions. Sometimes more than one receptor may be involved as well, and even multiple receptors can be used by the same virus. We are going to see these examples as well. And upon binding to the receptor, usually there is a dramatic conformational change that happens into the surface protein of the virus as well as in the receptor. Both of these proteins undergo a dramatic conformational change almost all the time. And that can trigger a lot of different things. It could be receptor-mediated endocytosis, that they deliver the virion inside the cell without uncoating, or at the cell surface, some uncoating can occur and it can deliver a subviral particle, which is a partial capsid, or it could be the nucleocapsid, many different types. And we're going to see that. But here the main point is that dramatic conformational change of the receptor is the most important part. So the cellular receptor for the viruses, except for those of fungi and the plants, enter up. So basically what happens that you need a cellular receptor for all the animal viruses. But the exceptions are the fungi and the plants. They have really tough cell walls, and also it's very difficult for them, the viruses, to penetrate. So usually in the plants, they enter through the wounds and then go from cell to cell as the cell divide, and the same thing for the fungus. They are always intracell. Due to a mechanical injury, they can enter the cell initially, and then can they travel from cell to cell as the cells divide. They cannot penetrate the cell just like animal cells with the help of a virus. See that in 1985, there is only one receptor was discovered at that time, sialic acid with the influenza virus. And then these were, like in the last 20, 30 years, a complete breakthrough in the field actually basically happened. I used to work with the vesicular stomatitis virus in the early 2000 at that time. Receptor was not known at that time. And now we know it's a low-density lipoprotein receptor. So it just discovered in the late 2010s, after 2010. So here are some of the enabling technologies for the discoveries, and we're just trying to see that how we discover these receptors. It is an extremely tedious task once upon a time. Now it has become easier due to this state-of-the-art technologies. And some of them you are familiar with, some of them you're not. Just do a little bit of your own background study using some internet resources like Wiki or something. It will help you. So first is a recombinant DNA technology. So you basically know that you just basically can clone a receptor, and that is great. I mean, like you just, if you're suspecting that a surface molecule on the cell surface, say there are 1,500 approximately on an average cell, animal cell or a human cell, there's about 1,500 proteins in the plasma membrane. And you suspect that, okay, all right, this may be the receptor. You just suspect it. You just clone that, produce that in vitro or in the cell or through the bacteria. You purify the protein and just study that. Very, very easy to do that. Then another thing what you can do is a monoclonal antibodies. If you basically think that, okay, all right, this protein A is the receptor, and what a monoclonal antibody does, that suppose it's a protein A, monoclonal antibody recognizes a single epitope. That means a single stretch of amino acid in a protein. So a single protein, if it's a surface protein, it will have many epitopes. There are several antibody binding sites. A monoclonal will understand only one of those several. So you can just have a monoclonal antibody against a specific site. You added those monoclonal antibody in a cell culture. So those monoclonal antibodies will bind to these specific sites. Now you challenge the cells with a virus. If you see that the virus cannot get into the cell, that's epitope or that is your receptor. This is the kind of study what you can do. Flow cytometry, you can just modify this experiment. Suppose you have those monoclonal antibodies, many different epitopes or regular antibodies bind to cell surface proteins, and suppose you have a protein A that you're suspecting, then you have monoclonal antibody A, monoclonal antibody B against the same protein, CD, something like that, and you label them with different colors. You have a green, red, yellow. You just label them with the fluorophores, these antibodies, and then you sort these cells with the flow cytometry. So after that, you challenge them with the virus and see that out of these cells, which one is preventing the viral infection, that virus cannot go in. That is, and just if you see that that's the yellow cell, that is where the virus infection is preventing, that particular epitope or the receptor is your receptor. That's what you can do. Of course, you can do the nucleotide sequencing of the receptors as well. So you can look for all the genomes. You can just look at all the proteins that is in the plasma membrane. You can look at their nucleotide sequences, and then what you can do that you can just compare that from a known database that which could be your potential protein that can act as a receptor. I mean, like these are many of these bioinformatics studies can lead you to narrow down the receptors. So 1,500. From there, you may narrow down it to 200. And then you do more studies to narrow them down. Sequencing can do this. Then the siRNA, which is a CRISPR-Cas9, these are the two technologies which exploded in the last decade. So probably in the street, everybody knows this siRNA, which is a silencing of a gene technology. CRISPR-Cas9 is the same thing, that you just design a guide RNA, you just deliver it to the cell, and it will basically bind to the specific RNA and chop it off, so you can silence the gene. So basically, just do a background study if you're not familiar with just in a few lines that what are those. But siRNA or the CRISPR-Cas9 basically can silence a gene in a cell. So now what you can do, that you can just buy commercially a genome-wide siRNA or CRISPR library. That means that in that library, you have every siRNA for every open reading frame of the genome multiple times, every gene. CRISPR, same thing. You have the same guide RNA for every open reading frame multiple times in there. So what you do that from this library, you clone this library into a viral vector. And each of these viral vector will have just one piece of this. Now you have the library in these viruses. Each one of them is unique with one of these siRNA and one of these guide RNA for the CRISPR-Cas9. You infect the cells with a very low MOI. So very low, remember that. This is all in your blurb. At that time, why you do a low MOI? Because you want just only one virus particle to infect one cell, not multiple. And you assume that if that theoretically happens, in each cell, one gene will be silenced. One gene, that's it. Now you challenge that whole thing. This is happening that you in the same experiment. You just challenge that virus after putting the guide RNA or the siRNA with your virus that you are studying and trying to look for the receptor. If you see that most of the cells will die, that means that they can enter the cell, they will kill the cell. It may happen that there's only one cell where the guide RNA or the siRNA silenced a receptor, and that cell will survive. So once all the cells are dead, that one is proliferating, you take the cell and see which gene you have silenced, that's your receptor. So these are some of the technologies which actually helped a lot for the viral discovery. So here is a lot of diverse cellular function. You can easily see that a lot of different receptor, and here you can see that we talked about in the very first slide that may involve passive molecules or receptors, and here it is. You can just see that here the attachment factors like heparan sulfate proteoglycan and the sialic acid is in the blue. These are attachment factors, means the sialic acid is found in almost every glycoprotein on the plasma membrane. So a lot of possibility for the influenza virus is using that to enter the cell. It would just specifically find to them and endocytose. So those are the attachment factors. On the other hand, here you can see these are specific receptors. And also in the GM1 ganglioside, see that the sialic acid is an important receptor. Remember that sialic acid is a class of alpha keto acid, 9-carbon backbone, and NANA, which is N-acetyl-neuramic acid, is one of the most predominant one. And that is the one which the influenza virus and most of the virus binds, again, in acetyl-neuramic acid or NANA. But for all practical purposes, we will just refer to their generic name, which is sialic acid. But remember that there are many types of sialic acids, just not only one, but that's what it is. So here you can see that this is an LDL receptor. This is called a DC sign. So I just joke with that. This is called a dendritic cell intracellular adhesion molecule grabbing 3 non-integrin. So I just say that because I'm just seeing this molecule so many times. I've read this so many times. That's what happens, that when you study more and more and over, you will just retain in your long-term memory. So that's basically what it is. You do not have to remember this name for your exam or quiz at all. Then CD4 and the CXCR5, these are, so here you have one receptor and a co-receptor for HIV-1. And also adenovirus also uses the CAR as well as the integrin. So you can just easily see that there are many different types of receptors and they come in all different shape and sizes. Just read the blurb. These are descriptive slides. So if the slide is descriptive like this, I'm not going to spend any time because we're going to spend more time where you need the concept or the mechanism. We're going to spend more time in there. So here is an example that here is a human herpes virus, herpes simplex virus. And see that, as we said, that one virus can use multiple receptors. So we can just see that right here. One virus can be use multiple receptors. A classic example, influenza virus. If a protein does have the sialic acid on the cell surface, that the influenza virus will bind many different types of protein. On the other hand, see, this virus have many different forms. You have a GP42, glycoprotein 42, most of them glycosylated, glycoprotein B, glycoprotein heavy or glycoprotein large, then glycoprotein D. These are all different types of proteins. And see that where they bind. Some of them bind to the heparan sulfate, some of them bind to the integrin, nectin, and herpes virus entry modulator. So all these different types of different molecules, they can bind to multiple receptors. So that's basically the concept, just not one. So here is just an idea that how the receptors bind onto the cell surface, and there are two examples in here. So first, let us see that what you are exactly looking at this picture. So on the left, you are seeing a poliovirus. The part which is gray is the soluble part of the receptor. And on the right is the rhinovirus. And see that this five-fold symmetry where you see that there is a gray part, this is the soluble part of the receptor. So what you do in these experiments, remember that the receptors is a cell surface protein. So it has a transmembrane domain because it is anchored into the plasma membrane. That transmembrane domain is extremely hydrophobic, and you cannot work with that because what happens that if you just express that, it will just immediately aggregate precipitate because you cannot work with that in an aqueous environment. So by recombinant DNA technology, what they do, the scientists basically express the ectodomain or the extracellular part of the receptor only, which sticks out of the membrane. This is in the environment, this is in the aqueous environment all the time, and you don't have any problem with that. Most of the time, these crystal structures, atomic structures are solved with that soluble extracellular domain of the receptor, and that's what you are seeing. First of all, the gray parts are the receptor, and there are two types that you are seeing. On the left-hand side, what you are seeing is a CD155 is a receptor, and see that this is binding into a pocket or a canyon. So this is the part which is in the canyon, which is inside like this. There is a pocket through which it is binding. On the other hand, what you are seeing right here in the rhinovirus, it is binding to a protruding surface. So there is a structure which is pretty much on the surface, no canyons, and the receptor can bind onto that. So depends upon that. You do not have to have a canyon. You can have a protruding surface or inside, depending upon where it fixed like a lock and key, that's what it is. So it comes in all different shapes and sizes. So here is another example of that. So the adenovirus does have this trimer shaft, like you can see, which is in here. You are looking from the threefold symmetry from the top. You have 1, 2, 3 molecules. Here you have 3 colored, the same thing. You are looking from the top. And at the bottom, you have this penton base, which has a 5-fold symmetry if you look from here. So the Coxsackie virus and adenovirus receptor, which is in the blue, see that where it is binding right here. So that is the first binding. But that first binding at the top right here in the knob is not sufficient for the virus to enter. You need another receptor interaction which happens in the penton base right here. The integrin co-receptor binds in here. You need both of them before the virus can be taken inside the cell. So this is a classic example that how this one is not enough. At the base of this symmetry, which is a 5-fold symmetry at the penton base, you need the integrin co-receptor binding for this as well. So here, on the other hand, the influenza virus attachment, we're going to see that the hemagglutinin is the protein that we already talked about that binds to these cell surface proteins having a self-sialic acid. There are many. And what you have in here, there is also another protein N, which is the neuraminidase. Now remember that the HA, hemagglutinin, in virology we say HA, but this is your H, H1, H2, H3, H5, that's it. Your neuraminidase is the N1, N2, N3, those are the N's. So the neuraminidase is not much in there. So here is an interesting concept that I explained that in that why you have this neuraminidase in that. So what happens, this hemagglutinin, just understand that very carefully. The hemagglutinin is the glycoprotein on the surface of the virus. As the virus comes out or the bud out of the cell, there are many glycoproteins on hemagglutinin which are glycosylated and have sialic acid. Again, repeating, the H-hemagglutinin does have lots of sialic acid on its surface as a glycoprotein. So what happens as the virus comes out of the cell, the hemagglutinin starts sticking to not only the cell, but others. Remember that hemagglutinin binds to sialic acid and the sialic acid present on the protein as well. So what will happen, the other hemagglutinin from the other virus is sticking somewhere to this, and it just becomes a mess. The viruses will aggregate. So the neuraminidase actually cleaves these sialic acid, so these viruses can spread out from the cell from these non-specific sialic acid binding, and that's the reason. And we saw that in a large high concentration of 1:4 in the hemagglutinin test, the sialic acid is cleaved by the neuraminidase, so that's the reason where the agglutination doesn't happen. So that's the function. So let's see that what is happening in here with the influenza virus. The sialic acid is present at the very top, and you can see that this is the terminal sugar, that's sialic acid, and it's attached to the second sugar, which is a galactose in a proteoglycan. And this linkage is usually a 2,3 linkage or a 2,6 linkage. So there are two different types way that the terminal sialic acid is bound to the glycose. Remember that this is the protein, this is your carbohydrate glycoprotein chain. The first one at the top, which is the yellow, is the sialic acid, and the second one's the galactose. Could be, and after that it could be different sugars as well. So very interestingly, the avian strains of the influenza virus recognize that 2,3 linkage, whereas the human strains prefer the 2,6 linkage. Now, this is the most important striking feature that what happened, that the H5N1 strain, which is the 1980 pandemic strain, that was essentially an avian strain. That was not being circulated into the human being. It was detrimental to the waterboards, which is the basic reservoir for the influenza virus. Now, what happened, that H5N1 strain had a mutation in a particular site where instead of recognizing the 2,3, which is the avian influenza, which the virus was from, it could now recognize the 2,6 linkage. Now that avian strain became a human strain, and it has not been seen by the human beings, and it caused that pandemic. So you can just really imagine that just one amino acid change can change the specificity from 2,3 to 2,6 and just cause these issues. Unbelievable. So here is the monomer of the hemagglutinin. Hemagglutinin, it remains as a trimer. Most of these are the trimers on the surface of the influenza virus, and you are looking at just one monomer. This is the globular head, and this is a stalk portion, and we call this portions as HA1 and the HA2. And right here, which is not shown, there will be a transmembrane domain which is inserted into the membrane, and this will be the viral envelope of the membrane, and it sticks out from there. And this is the crystal structure. This has been shown here. And here you can see that this is the globular head. On top of this right here, there is a sialic acid binding pocket, and the sialic acid is bound, and you have 1, 2, 3, 4, 5 hydrogen bondings with the sialic acid and that. So now you understand why I said that interaction with the receptor is multivalent. This is what multivalent means. You have just not one hydrogen bonding. That will be too weak. You have 5 hydrogen bonding in there, and the receptor is locked in onto the HA globular trimer. And that's what we mean by the multivalent interaction. So here is also another example of HIV-1. And the HIV-1 has 2 proteins which is produced, which is called the SU, surface protein, and the stalk is called the TM, or the transmembrane. And here, again, the transmembrane is difficult to work with, so we are here working with the surface protein. So they are also known as GP120, the surface protein. The 120 is 120 kilodalton is the molecular weight. GP41 is a transmembrane protein, is 41 kilodalton molecular weight. So that's what it is. And you can see that cryo-electron micrograph and the tomographic image. Here, what you are seeing, that on the top, this is the surface protein right here. And this is the CD4, which is the receptor that you find on the macrophages or on the T cells. And it contains that what you are seeing with, that sticking out, that aromatic ring, that is an that is the hydrophobic amino acid, it's phenylalanine. And that phenylalanine, that just single amino acid, is the most important one for the receptor binding interaction. If you just take those that phenylalanine out, you are not going to get any binding in there. So you can just so it's a phenylalanine at the 43rd position. It is so important in there. So this is basically the idea. So remember that when a virus is interacting with the receptor, the types of interactions are just not electrostatic. Electrostatic is basically how the virus just comes onto the cell surface and randomly attach. These interactions, we saw that right here, hydrogen bonding. Here, the interaction is with an aromatic ring of an amino acid, so it's a hydrophobic protein-protein interaction. This is a second type of interactions. Along with that, it could be van der Waals force as well. So these are the different interactions. On top of that, electrostatics may help as well. But mainly remember that the receptor and the surface protein interaction of the virus is usually hydrogen bonding, hydrophobic interaction, and van der Waals. These are all in your blurb. So these are the different types of things that the cell uses to get material inside. So this is phagocytosis, you know very well. This is not the typical mode of entry of the virus into the cell. This can be done by the macrophages. This can be done by many of the neutrophils, but that is usually not the main way of the virus. Randomly, it can take sometimes. The dendritic cells and the macrophages can do that, take up virus particles by phagocytosis, but that's not for the infection purposes. At that time, these dendritic cells and the macrophages, they are called the antigen-presenting cells. They capture and present it to the immune system for different functions. That's not for the infection purpose as well. So this is not a mode of entry for regular viruses. On the other hand, macropinocytosis, also known as the fluid phase phagocytosis or fluid phase endocytosis, small molecules and liquid are taking up. Some of the small viruses, due to this by membrane ruffling, they can get into the cells. But again, these are not the main entry mechanism for the viruses. Whereas the receptor-mediated endocytosis is usually considered as the most important and most predominant type of. You need a ligand. This ligand is a viral surface protein and doesn't have to be a protein that is sticking out. We're going to see that you have a naked capsid that can also bind to a receptor as well. And that's after that how you take it inside. So the cytoplasm is very crowded. This is a classic picture of that. You can see that these are the glycoproteins in there. These are the cytoskeletal structure. You can easily see this is the histones in the nucleus. You can see that how they are. You can see the ribosomes and the endoplasmic reticulums is a very classic picture. This is just for conceptual purpose, but it's very crowded. So how the virus actually travels inside the cell is fascinating. That's what we are going to see. It's a busy slide, so let's go from one side to the other. So this is also written in the barb. So first we are seeing the A1, where you can see that this virus is getting uncoated at the plasma membrane. So here it binds to the receptor and fuses with the plasma membrane, and the nucleocapsid is delivered into the cell. Measles virus actually do that. But just know the mechanism. You do not have to remember each and every example of these though. The second one, on the other hand, you can see that this is a naked virus. And with the receptor, it can just directly enter into the cell. And then what you are seeing, that this virus need to get into its genome into the nucleus, for example. Sometimes it may be just delivering to the cytoplasm. So what you are seeing, that right here is a microtubule. This is the centrosome, which is also called the microtubule organization center. And the microtubules are the cytoskeleton, which is radiating out all throughout. Near the microtubule organization center, or the centrosome, is the minus end of the microtubule. Towards the periphery, it is the plus end of the microtubule. So you have specific motors which travel on these microtubules carrying cargo. Here, the cargo or the motor protein which is carried by is the dynein. Dynein is the motor protein which goes from the plus end towards the minus end, towards the center of the cell. And then what happens? Now it goes all the way to the cell, but it did not reach the nucleus as well. So it just moves. Now it's moving from the minus towards the plus end. And this is by kinesin, which moves. Kinesin is also the motor protein, which moves from the minus to the plus end. As it is moving, it will find a nuclear pore and it will dock into the nuclear pore. And the kinesin will still try to pull it apart and it just and deliver the nucleic acid. It will just, the whole structure will come apart. We're going to see that. So in this process, on the other hand, that the viruses actually bind to different types of receptors, and these receptors are constantly formed in pits, these receptors are taken inside the cell through these clathrin-coated pits. Now remember that these clathrin-coated pits are constantly being formed and recycled onto the cell surface. They are constantly pinched off and recycled, pinched off and recycled. Now, if the receptor along with the virus finds itself in the clathrin-coated pit, it will be taken in, and the pinching off is done by a protein which is called the dynamin. So definitely underline that protein, dynamin, which pinches off the vesicle, and this clathrin-coated vesicle is now being carried the same way on the dynein, and it will meet or it will transform into an endosome. So what happens in the endosome, a lot of different things. The endosome do have proton pumps, and it's going to pump protons, H+, from the cytoplasm into the endosome, and it becomes extremely acidic. This acidity will change the conformation of different proteins, and the capsid or the nucleocapsid will come apart, and the genome is delivered into the cytoplasm. And then it may be transported after that to the nucleus as well. Very important thing, when there is a plasma membrane fusion, this is happening at neutral pH. Remember that at the plasma membrane, the fusion is at the neutral pH. When the endosome is being, inside the endosome, when the virus is being fused with the endosome, this happens in the acidic pH. So very different. The other types of things can happen, that which is a clathrin or caveolin independent internalization of the virus. Here also, there is a receptor that is needed, and that will internalize the viral particle as well. So what is a caveolin? Caveolin is another structure which forms a flask-shaped structure. It is not formed by the clathrin. Clathrin does not form these pits, but these pits are formed by the caveolin 1. There are many others. And some of the virus can bind to receptors which are taken in by the caveolin-coated vesicles. And it forms in a caveosome. Now remember that, very different in the caveosome. From the caveosome, sometimes some of the virus goes into the endoplasmic reticulum. This is a pH neutral route. This is, if they are not in an endosome, it is not in an acidic environment. It's a pH neutral route. So this is also another important way the viruses can get inside the cell as well. So this is I'm just going to shortly play that because there are many videos and you can just all play those. This is a typical structure of a kinesin carrying a vesicle which may contain a virus inside that. And these are based on the molecular structure of the kinesin and the microtubules. So it is cartoon-like, but the structure is, each of these conformations are crystallographically correct. So it just literally walks with the power of ATP on the microtubules. All right, so after that, we are looking at this at the neutral pH, what is happening. So there are two examples, measles virus as well as the HIV. So the measles virus, we are looking at a structure. This is the viral membrane, and there are two of these proteins. And you can see that this is a hemagglutinin protein, and this structure right here is a fusion protein. And very interestingly, what happens that if you can see that this fusion protein is connected still, this is just one protein, very carefully see that, connected by a stretch of peptide, which looks like that, but F1, F2 is a single polypeptide as the virus is coming out of the cell. F1, F2 is a single polypeptide. We call it an F0 usually, single polypeptide. Now, as the virus is coming out of the cell, see that the first two picture we are talking about when the virus is being manufactured. Third picture is onwards is the entry. There are lots of cell surface proteases. All the cell surface have lots of these proteases. As the virus is assembling and coming out of the cell, these cell surface proteases cleave this protein into two parts. Now, in reality, this part we call as an F1, and this becomes the F2. And see that right here, this is not attached anymore, but the proteins are still attached by a disulfide bond. And the fusion peptide remains, remember that the fusion protein will be inserted into the membrane. And one of the most important character of the fusion peptide is that it is a stretch of hydrophobic amino acid. Again, remember that the fusion peptide is a stretch of hydrophobic amino acid, extremely unstable in aqueous environment. So that fusion peptide needed to be hidden in a pocket onto the viral surface. So here you can see that it is cleaved, but that fusion peptide is now may see the aqueous environment, but that's the reason that it is still in that pocket, and the two structures are connected by the disulfide bond. So now this is the viral receptor is now binding to the viral surface protein, and now it will have a dramatic conformational change to the HN, and that dramatic conformational change will convey due to that, this fusion peptide will now from here flip out due to the conformational change, flip out and insert these hydrophobic part into the membrane. And then we are going to see that there's some conformational change will pull the two membranes together to fuse. Again, remember that conformational change took that fusion peptide, flipped that fusion peptide out to insert that. Same thing. For the HIV, you have an ECU and the transmembrane domain, and you have the fusion peptide which is hidden into a pocket in an HIV. When it binds to the CD4 along with the co-receptor CCR5 or CXCR4, depending upon what it is, that allows that fusion peptide to flip out and insert into the membrane. That will cause the fusion. And we're going to see that this happens in the neutral pH. Highly regulated, you can easily see very specific receptor interactions are required to happen this in neutral pH. So this is just a little bit of this, and I'm just going to fast forward this because otherwise it will take. So this is your HIV. Okay. So here we are looking at, this is right here is the SU and the TM, GP41 and GP120. It's now you can see that this is the most important part that if you very carefully see that. After binding, you will see the green one, GP41, would flip its fusion peptide that will be inserted into the membrane. So just look at that. That is a very subtle, but that is the most important part. So right here. Right there, the fusion peptide. And now, see that what happens. This will cause a conformational change by automatic hairpinning. We call this hairpinning. And the two membranes, this is phospholipid bilayer, will come together, and the virus will basically have a free passage for the nucleocapsid that will be delivered inside the cell. So this is the basic mechanism of a fusion and delivery of the genome. So that's it. So this is the essentially that how it goes for most of the thing. And again, here it is just shows the hairpinning in a much more elaborate way that how this happens. And again, I'm just not going to go into the whole detail of that because we will just run out of time. So we already saw the fusion. Let's see right here. So see the fusion peptides are inserted, and this is the hairpinning purpose. The viral as well as the two membranes come out due to the conformational change of these proteins, and you have a free passage in there. And once you have the free passage, this is the free passage. The material from the virus can be delivered inside. So you can just take a look at these at your leisure. So here is the specific thing that is happening at endosomal membrane in acidic pH, and this is an influenza virus entry. So you have the hemagglutinin. It ends up in the endosome after you have the receptor-mediated endocytosis. And these are the proton pumps on the endosomal membrane which will pump the protons. And upon the proton pumping, you can see that this will cause, again, after, now very carefully see that. Now the endosome is full of these protons. On the membrane, other than H and N, there is also another protein that we never talked about. This is called an M2 protein. This is a proton pump on the membrane of the virus itself. It's a proton pump. That proton pump will now take in the H plus or the proton from the endosome towards the inside of this. And that will cause the membrane fusion. Once the membrane is fused, you can see that these are the nucleocapsids, which are the 8 segments of RNA encapsulated with the protein. They are released into the cytoplasm, and you can see that in this process, this M1 protein also is involved in this process. And these nucleocapsids are used, they are transported into the nucleus by some specific import proteins. So import proteins are basically catch the proteins that is supposed to go into the nucleus. They have a signature sequence. The import proteins find them, and they are transported through the nuclear pore. So here what you are looking at, that we are looking at the trimer of the hemagglutinin, and see that the yellow part is the folded up part of the hemagglutinin that we actually saw. So this protein is nothing but a trimer of this. That's what we are seeing right here. This is the same protein. All right. And now see that what is happening, that it is loosened up in the endosome. And at this point, when it is loosened up, it flips this portion right here. This portion has the buried fusion peptide that you cannot see. This flipped out towards the membrane, and this red part is the fusion peptide. And this will cause the hairpinning process, and the two membranes will basically fuse together. So that is exactly what is happening right here. So this is the membrane fusion by the H or the hemagglutinin fusion protein. All right, so here is just also another crystal structure. So what is happening right here? This is the globular head. This is the rod-shaped structure of that. And in this hydrophobic cavity, the fusion peptide is right here. So this is called the H0, and this protein is present on the virus as it is coming out. As I said, that the host cell has all these cell membrane proteases. These proteases will basically cleave this, and now you have a new H, N, and C terminal. You have an H1 and H2. So you have an HA0. From that, you have an HA1 and an HA2, or H1 or H2. Both are correct. So now you have this fusion peptide right here, and see that this is the conformation in the neutral pH. But what happens that this whole side of this, that here you have the N and the C terminal right here, this whole thing is flipped towards the other side in the acidic pH. And again, if you're not in structural biology, sometimes it is difficult to understand that. So the fusion peptide is in the hydrophobic cavity after the cleavage, and it is flipped towards the endosomal membrane in acidic pH. That's what we saw in the previous slide, basically. At the molecular level, that's what it is. All right, so you can just look at this is a pretty long video, and you can just, it's a 4-minute, and we'll just run out of time, but you can just look at the animation. These are some of the cool animations that you can easily see, will give you a much more clear perspective of that. So now we have already talked about this protein, and look at this fusion proteins, which we call as a class 1 fusion protein. And we were talking about the influenza virus. All these are class 1 fusion proteins. which have a specific architecture. They are perpendicular to the membrane and looks like a spike. They are mostly alpha helical. You can see that mostly alpha helical and form trimers. You have 1, 2, 3. You have 1, 2, 3 monomers together and they are trimer. So you have a stalk and a globular head, and in the middle of the globular head is usually the receptor binding site. You can see in the simian virus 5, in Ebola, HIV, murine leukemia virus, this is a retrovirus as well. How strikingly these spike proteins are. They are very, very similar to these, and we're going to see that it will be the similar for your SARS-CoV-2 as well. The spike protein will be a trimer. It will look like this. It's a class 1 fusion protein. So just know that what are the class 1 fusion proteins? These are perpendicular to the membrane, mostly alpha helicals, and form trimers. They are sticking out like bristles out of the viruses. Many of the viruses use that, but not all. So the second type of class 2 fusion protein is interesting, that right here what is happening is these are mostly beta sheets and form dimers, and they are parallel to the membrane. So if you just look at lots of flaviviruses does that. So flaviviruses include dengue, yellow fever, Zika, Japanese encephalitis. These are all flaviviruses. Some of them are really dangerous. And you can see that this is a 5-fold symmetry. You can see that how these proteins are flat, they are not sticking out. And here you can just easily see that this is a 3-fold symmetry, 1, 2, and 3. Around the red, they're 3-fold symmetry. Here is a 5-fold symmetry. So they are flat. And see that how the mechanism works, that in the virus, this protein lays flat on the surface of the virus, but basically it's the same thing. When there is a proper condition and binding to the receptor, they just flip towards the membrane, and the hairpinning process will bring the two membranes together, and you have a free passage in between the virus and the cell. It is the same mechanism. So it does not matter whether the protein is sticking out or flat on the surface. Everything, the mechanism of entry or the membrane fusion is basically the same. How you just bring the two phospholipid membranes that they become just one for a free passage. And here you can, of course, take a look at the animation, and we are not, it's a pretty long animation. Then we're not going to spend that much, or probably not. That. So the two membranes is basically the same thing in there. So you can just all take a look at on your own of this. Now, after that, we are also looking at another example of a low pH mediated mechanism, and this happens in the Ebola virus. And this is very, very interesting. So the Ebola virus do have a glycoprotein on its surface, and first is enter the cell. Now, think about this. Still, we do not know the first step. We do not know the receptor on the cell, that how it enters. It just ends up in the endosome by receptor-mediated endocytosis, but we still do not know the receptor. So that's what the interesting part is. But from the endosome, we know the pathway. So right here, what happens that in the acidic pH right here, you can see that this glycoprotein on the top of this, you can see that there is a dot, a magenta dot at the top. And this is called, there is a glycan or mucin cap on top of that. So they're capped. So in the endosome, first the hydrogen ions or the protons are pumped. And then in the very late endosome, many of the viruses actually in the pathway, they escape before the endosome is fused with the lysosome. So again, just in a short, if you just recap the cell biology, an endosome is formed with the material that is coming into the virus, coming into the cell. First, what happened? The acidity of the there is a proton gets into the endosome, and the pH becomes acidic. That's the first part, late endosome. Towards the very late endosome, the lysosome will fuse with the endosome, and all the lysosomal proteins, the proteases, nucleases, these are all acid proteases, means that these are proteases or the nucleases, hydrolases, they all work in acidic pH. So what happens that in the acidic pH, the lysosomal proteases, they cleave that glycoprotein, and see that this is Cl, that is the glycoprotein is cleaved, that the mucin cap is gone. Mucin or the glycan cap is gone. At that time, it can bind to its receptor on the surface of the endosome, which is called the NPC1, Niemann-Pick1 protein. And the interesting part is that what is the function of NPC1? This is actually responsible for low-density lipoprotein. It's an LDL receptor. So the people who doesn't have NPC1, they have problems with cholesterol metabolism. And interestingly, those people are resistant to Ebola. So that's how it was discovered that the people who doesn't express NPC1 are resistant to Ebola. It cannot get into the cell. So this is also another interesting thing where you need a proteolytic cleavage inside the endosome. So again, this is a recap slide. Fusion is highly regulated, must not occur in the wrong location. It happens in the neutral pH in plasma membrane, and in the low pH fusion, it could be a proteolytic cleavage activates the fusion protein for cleavage. This is class 1. Cleavage of a second protein, class 2, activates the fusion protein, and the endosome fusion receptor is also present that we saw in the Ebola. Now we are looking at that how the genomes are released from a non-enveloped virus. We're just always looking at the enveloped virus, which are with the spikes. Here also you have some fiber-like, and this is a typical structure that what you are seeing in an adenovirus. Right here you can see that right here is a fiber receptor, and you have a co-receptor integrin, and that will take the adenovirus inside the cell endosome, and the acidified endosome will partially degrade the viral capsid, partially degrade. And that partially degraded capsid will be delivered into the cytoplasm. We call this as a subviral particle. This is an important thing. It's a partially digested, it's not intact, partially digested subviral particle. It is not that robust as the intact virus. So after that, what happened, that this subviral particle is then carried by these dynein motors to the nuclear pore complex. And at the nuclear pore complex, it would deliver its genome, and we're going to see that how this will be again torn apart in that process to open up so that the genome can be delivered into the nucleus. We're going to see that. This is another one, the poliovirus, which is intact, doesn't even have the spikes. It's just an icosahedral virus with a completely plain surface. On the top, nothing sticks out. And how it happens. Still, it binds to a specific receptor, which is a CD155. That is the receptor. And we're going to see that there is a cation on the virus that is bound. And it is, again, comes into the endosome from there. This is partially degraded, and we call this one as an altered ape or the altered particle. So there is some specific interactions goes on that changes the conformation of this virus, and the genome is delivered into the cytoplasm here. It's a poliovirus, it goes into the cytoplasm. So what happens really here? We are looking at the site through which the genome goes into the cell. So here you have the particular protein, and you can see that there is a protein called VP3, then you have the VP4, you have the VP1. Right here, you have a pocket right here, and this pocket contains a lipid. Very interestingly, the black part is a lipid. It's usually sphingosine. This is a cellular lipid. So what happens that when the virus is actually manufactured, that lipid is put into the place. If the lipid is not in there, the virus will not mature. It will not be able to keep its genome intact. This is what we call that. Think about this way, that lipid actually goes in there and spring loads the virus. Remember that the virus has to be a metastable structure. And this is, think about that the structure is a little bit loose. You put the lipid in there and it becomes tight. And if the lipid goes out, it again becomes loose to deliver that. This is your spring-loaded structure that we talked about at the very early, which gives the metastability of the virion. So this is spring-loaded. Now what happens that when the receptor CD155 binds to this, there is a conformational change which releases the spring or the lipid. The lipid is extruded out from the pocket. As the lipid comes out, think the tension of the spring, just analogy, changes the whole conformation of this VP1, VP2, VP4, and they flip out and insert into the membrane. And they make a free passage through which the RNA goes into the cell. And it is connected with the VPG, that is a protein, though we're not going to need it. But that's basically what it is. You can see that right here, the VP3 was the plug, which is kept in the space. The RNA cannot go in. As the VP1 and the VP4 is inserted into the membrane, that removes that plug of the VP3, and now the RNA can go inside the cytoplasm. That's the delivery. So here is a structure right here. You can see CD55 is bound into the canyon, and below the canyon is that structure right here where you have the lipid actually stays in here. So what you are seeing in this structure, this is a compound which is known as a Wnt compound. This was an antiviral compound that was developed by the Sterling Went company long time ago. And this is actually an antiviral for the poliovirus. But why it is not again used? Resistance grows very quickly against that, and it could specifically remove that Wnt compound as well. So here you can see that instead of the lipid, if you put this compound in there, this will sit in there, and this will not allow the lipid to be there, and the virus will not be spring-loaded. So basically, the virus will not be infectious without that lipid. So this is basically a competitive analog of the lipid, which will have the antiviral property. But again, it did not work though that well. Here is another very interesting way that uncoding in the lysosome, what happens in the reovirus. We talked about the reovirus has a double capsid. The outer capsid is T equal to 13. The inside is T equal to 2. Not needed here. You have seen that somewhere else. So here what is happening, that by receptor-mediated endocytosis, this reovirus will get into the endosome. And in the endosome, the lysosome will fuse, and it will undergo the proteolysis. So the outer coat, which is the purple part, you can see that purple and the red part, that is digested, and the core comes out. The core actually contains some of these. So this is the core actually do contain some of these hydrophobic protein residues that cannot be on the surface. So remember that. This is what it is, why it is a double coat. This is the virion. Inside that you have this inside particle. On the surface of that you have some of the hydrophobic proteins which can fuse with the membrane. This will be completely unstable as a virus. So the virus is first digest this outer part, and this is your intermediate subviral particle and then once this is digested, the core actually comes out into the cytoplasm. The core remains in the cytoplasm. Interestingly, the RNA, genome RNA, is not delivered into the cytoplasm. For the reovirus, the RNA-dependent RNA polymerase is inside the virus, and the mRNAs actually come out from those tips. So the mRNA is delivered from here. The genome RNA is never delivered from here. So this is also another very interesting phenomenon. So what is basically the role of the co-receptor in the viral infection? And this is a very interesting thing, that the entry of the Coxsackie virus group V requires 2 receptors. And this is true for some of the adenoviruses as well. One is called a DAF, decay accelerating factor, or CD55, and the CAR, which is the Coxsackie virus adenovirus receptor. This is the same receptor for all. So why this happened, that suppose if you have these cells right here, in between the cells, you have these tight junctions. These are the tight junctions in between the cell. The CAR is a component of the tight junctions, and the virus really cannot go into that place because it is so tight, the virus don't have access to the CAR. So how the virus will basically go in there and use the CAR as a receptor? They will use the decay accelerating factor or the DAF or CD55 to change the cell surface on the side or the lateral side of the cell, modulate the cell surface so that the CARs are exposed. And that's what we are going to see in the next slide. So here you can see that suppose this is an epithelial cell, and right here the DAF is on a small lipid raft. You know, the lipid rafts are microdomains on the plasma membrane which concentrate a particular type of protein. And in this right here, the virus binds to the DAF or the CD to the DAF basically. This binding will undergo a full signal transduction pathway, and due to this signal transduction pathway, this is actin remodeling, which will basically remodel the tight junction and open it up. See that it was earlier completely at tight junctions and the two cells were completely sticking together. There is no way that the virus can enter in there and access the CAR receptor. But binding to the DAF will undergo a remodeling of the actin right here. It will open up the lateral surface of the cell, and the CAR is now exposed through which the virus can go in along with that. In adenovirus, it will use the CAR and the integrin as well on the on this surface, and it will just be delivered where it needs to go. So here, on the other hand, what we are looking at the 4 modes of nuclear entry. So this is a typical structure of the influenza virus, which the nucleocapsids are released from the endosome and then imported into the nucleus through the nuclear pore. Some of the viruses can directly go to the nucleus, like parvoviruses, small viruses can directly go to the nuclear pore, and conformational changes can deliver the nucleic acid in there. There are some cases where a subviral particle is formed. We talked about that partially digested, say, adenovirus, once they are in there, upon binding, they can deliver the nucleic acid. And in some of the viruses which are really small, like calcivirus or the parvovirus, the whole particle has the ability to go through the nuclear pore and deliver the nucleic acid into the nucleus. So here is one of the molecular mechanism that at the molecular scale that can happen to B as well as a C. You can see that these can all happen in here. And what is the mechanism? So this is right here is the dynein and the kinesin on the same viral particle. Dynein brings this molecule right in here. Now see, now as it flips over, kinesin is now attaching onto the microtubule. So one end of this capsid is attached to the nuclear pore very tightly. Now the kinesin wants to go in the opposite direction. Remember that dynein actually goes in, the dynein movement is called the retrograde. It goes towards the inside of the cell. Whereas the kinesin movement is anterograde. It just moves away from the cell. So it can just, or from the middle of the cell. So you can see that the other protein can now just pull it apart, and in that process, the whole capsid actually is broken down. You can easily see, and the nucleic acid is delivered. And the transportin is usually just a generic name of import proteins. They can recognize particular protein signals in different amino acid sequences in all these different proteins. When they bind to those proteins, they are ready to be destined into the nucleus, and that's how the nucleus, the material is delivered. So that's pretty much about it. So we never finish in time without rushing a little bit. So that's why I decided to skip over the videos a little bit, because those videos, those videos altogether is probably close to 10 minutes. So that's the reason that we did. So pretty much that's about it. So any questions? And anything in general, any questions that you may have? Anything? So just make sure that I will just upload, open the quiz. So you will have 2 of the quizzes. So don't wait until the end of the week, because both the structure is quite a lot. So try to do the structure, the viral structure quiz a little bit early, so that you have a little bit of time, and you know that we have a home game on Saturday. So lots of busy schedule. So just to remember that as well. So what do you guys think? We never have extra time to talk. So what do you think last week? Defense was it. I mean, like, it's unbelievable that you do not have an offensive touchdown. But what do you think? That do you think that our offense is going to turn around? I mean, like, I'm just a little bit worried about the NC State, and though that's an away game, and the Syracuse game. Hopefully, the other ones I'm not, but I'm just trying to keep my fingers crossed. But this was not an elimination game, so we're just still pretty much in the game. But we'll see. What? Yeah, so hopefully defense is great. But I'm like, this is one of the things that you know Skalski personally. Anyone? James Skalski? Anyone? Why he puts his head down every time he tackles? How many times he got ejected? This time also it was not really, but he always puts his head down. I was like, well, I'm not a Dabu or trying to be one to tell him that, but that's one of the things that what I see, this he always puts his head down, and that just like gets into trouble in bigger games where he still have a bad call. Do you see that in that punt tackle, and they make it up with another kind of a iffy call. Anyway. So we'll talk about, so we'll do the review on Thursday. Honor students, you have a lot to do this weekend, by this weekend, so just try to do early. All right, and again, please reach out for one-to-one if you need to go over the material. I can't emphasize that enough. All right, we'll see you Thursday.